This is an introduction to the process of transcription. Before learning about transcription, you will need to have learned about cell organelles, the structure of DNA, and the process of DNA replication. Once you have an understanding of those things, you can begin learning about transcription. A big part of a cell's job is to make proteins. Bodies need proteins for physical structures such as part of the cell's membrane, and for muscles, and for enzymes like lactase, which helps us digest lactose, the sugar in milk. Cells make proteins by using the instructions or codes contained in the DNA. Remember, DNA exists in the nucleus of cells and is a double-stranded molecule that consists of sequences of nucleotides. These nucleotides contain paired bases of A's and T's and G's and C's. A is always paired to T, and the same is true of G with C. The order of the nucleotides is what determines the type of protein that will be made. Because DNA cannot leave the nucleus of the cell, it needs a way to get its information out of the nucleus and into the cytoplasm of the cell, where the proteins are made. It does this through a process called transcription. Transcription is the copying of the code or nucleotide sequence from DNA inside the nucleus. Transcription uses one strand of DNA to make a copy of its nucleotide sequence when DNA is unzipped. An enzyme assembles pieces of nucleotides called messenger RNA, or mRNA for short, using the strand of DNA as a template. Complement bases of messenger RNA bond with the bases in the unzipped section of DNA, very similar to DNA replication. So a messenger RNA A will form a bond with the DNase T. A messenger RNA G will bond with the DNase C. Once a section is complete, the messenger RNA will break off from the DNA template, then will pass through the nuclear membrane or nuclear envelope, and make its way to the organelle called the rough endoplasmic reticulum, or rough ER, which contains ribosomes. It is at the ribosomes that proteins are assembled. Now is a good time to talk about the differences between DNA and RNA. RNA is very similar to DNA. It is a nucleic acid just as DNA, made of a base, a sugar, and a phosphate group, but there are a few differences. First, the nitrogenous base T found in DNA does not exist in RNA. Instead, there is a U, which stands for uracil, which bonds with A. Second, DNA exists as a twisted double strand, but RNA is single-stranded. Lastly, there is only one type of DNA, but there are three types of RNA: mRNA, or messenger RNA, tRNA, or transfer RNA, and rRNA, or ribosomal RNA, which are actually the ribosomes. By the way, DNA is named after the sugar in its nucleotides, deoxyribose. RNA is named after its sugar, which is ribose. So messenger RNA is how the DNA code gets from the nucleus of the cell into the cytoplasm. Once in the cytoplasm, the messenger RNA makes its way to the ribosomes on the rough ER. In a sense, the DNA hands off its message to a strand of messenger RNA. So let's begin the process of transcription at the unzipped DNA back in the nucleus to make a segment of messenger RNA. If the DNA sequence is A, G, G, T, which messenger RNA bases will bond with each DNA base? Pause this lesson here and write down the bases that complement DNA's A, G, G, T. If your answer is U, C, C, A, you are correct. Remember that there are no T's in RNA, so it's a U that complements the DNA's A. After each base pair bonds, the sugar and phosphate parts bond with the ones above and below them to begin making a strand. Time for another quick check. Pause this video and write down the three differences between DNA and RNA. If you wrote that DNA has a T base but RNA has a U, that DNA has two strands but RNA has one, and that there is only one kind of DNA but three types of RNA, you got it right. The type of RNA we haven't talked about yet, transfer RNA, is used next in the process called translation, which is the assembling of amino acids into proteins.